Today I will discuss about DNA replication. DNA replications is one of the most important matter. DNA is a molecule made up of two strands and twisted around each other in double helix shapes. DNA in here each strand is made up of sequence of four chemical bases such as represented by the letter A, C, G and T. A denotes for adenine, C cytosine, G guanine and T thymine. The two strands are complementary. This means that wherever there's a T in one strand, there will be in a in an opposite strand, and whereas there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end in the direction. The two strands runs in opposite direction, 5' to 3' and another 3' to 5' direction. That determines how each strand of DNA is replicated. The first steps in DNA replications is to separate the two strands. You also look in a video. This unzipping is done by an enzyme called helicase. Helicase is an enzyme and it results in the formation of a replication fork. This is a replication fork. You also look it. The separated strands each provided a template for creating a new strands of DNA. This is called template strand. An enzyme called primase starts the process. First of all, the enzymes is also attached with the DNA strands. This enzyme is called primase. This is RNA based. This enzyme make a small pieces of mRNA called primer. This is called primer. First of all, it is created. This marks the starting point for the construction of the new strands of DNA. An enzyme called DNA polymerase binds to the primer. This is a DNA polymerase and will make the new strands of DNA. DNA polymerase is an enzyme can only add DNA base in one direction from the 5' end to the 3' end. Look at my video. One of the new strands of DNA, the leading strand. These strands are produced. Leading strand. The DNA polymerase adding the base one by one in 5' to 3' direction. The other strands, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make these strands in a series of small chunks called Okazaki fragment. This is a Okazaki fragment. This is primase. Each fragment is studied with RNA primer. This is another strand of DNA. They produce a primer for the formation of DNA strands. DNA polymerase then added to short row of DNA waste. This enzyme is DNA polymerase in the direction 5' to 3' direction. This is attached of waste by RNA polymerase and repeated. The next primer is then added further down the lagging strands. Another Okazaki fragment is then made and the process is repeated again. These enzymes also help it DNA polymerase. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzymes then fills into gaps that are left behind to adhesions with the nitrogenous base with DNA strand. Now look at my video. Finally, the enzyme DNA ligase seals up the fragments of the DNA where the broken of the DNA strands. Ligase also fulfill in the both strands from a continuous double strands. DNA replications is described as semi-conservative, and after that they are coiling forming because each DNA molecule is made up of and one old conserved DNA strand. And also one new one strands they also make it. You also look my video. Thank you.